So last week we began discussing this last week we began discussing chapter 4, and that was genes and cellular function. We have realized that we began with the central dogma of biology, and from the central dogma of biology we learned that the directional flow of information is from the DNA made up of nucleotides into an RNA transcript that is made up of nucleotides, and then that transcription of the DNA into mRNA will be translated outside in the cytoplasm by the ribosomes, and these are translation machines that translate our mRNA transcript into our desired proteins that are structural proteins and functional proteins. It's very important for us to remember the difference between the structural proteins and the functional proteins. We talked about two types of structural proteins. One of them was keratin, excellent. And we will see in the tissue lecture about keratinized squamous cells that this keratin is very important. It's found in the hair cells as well as on the skin. What's the other form of, what's the name of the other structural protein? That will be collagen. Where do we usually find collagen? Collagen is found in the ligaments and the bone. And these structural proteins, they are there for tensile and structural support. There are other proteins that are known as, by the way, structural proteins also mean these leak channels, these transmembrane channels that open and allow for the passage, like tunnels, for the passage of these electrolytes down concentration gradient. These are types of structural proteins. And you have sodium potassium ATPases that are structural but also in the same time functional, because they have the function of causing and generating this disequilibrium, ensuring that any directional flow of sodium down concentration gradient from the outside to the inside is reversed, where we have 3 sodium ions sent outside the cell and 2 potassium sent inside the cell. And then we have other functional proteins, and these are always our enzymes. Our enzymes are a perfect example of what is a functional protein, like hemoglobin. Hemoglobin is a functional protein, it's a functional enzyme. It has the function of binding oxygen, transporting oxygen, and delivering oxygen, unloading oxygen, and then loading carbon dioxide, delivering it back into the lungs. That's an example of a functional protein. There are proteins that are functional in the sense that they neutralize reactive oxidative species. They are important for decreasing oxidative stress. They oxidize hydrogen peroxide. They mute the superoxide ions. We have other types of enzymes that are able to do functions. So not all the proteins are functional, not all the proteins are structural, but all the proteins are made up of amino acids, and all the amino acids were encoded by nucleotides, and they were translated in the ribosome. And we talked about our different macromolecules. We talked about our sugars. They are made up of monosaccharides, and then they come up as disaccharides, and then the disaccharides are added into polysaccharides. Examples of our monosaccharides will be our fructose, glucose, and galactose. Our disaccharides are the lactoses and the maltose and the sucrose. And then what's the example of the polysaccharides? What are the examples of our polysaccharides? Yes, starch, cellulose, and glycogen. Okay, and we also talked about the macromolecules, the fats, made up of glycerols and free fatty acids. And before that, we talked about the proteins made up of amino acids. Now we moved into the nucleotides. So the most important thing for us to know about the structure of a nucleotide is that it is a monomer. This nucleotide is a monomer, and many nucleotides give us a polymer. What do we call the polymer of nucleotides? Yeah, that's the nucleic acid. And the major example of nucleic acid would be the DNA and the RNA. So we have to have a very excellent understanding of the structure of all the micromolecules. So when we look at this structure before us right now, what is this? Is this a nucleotide or is it a nucleoside? That is a nucleotide. How do we tell the difference between a nucleotide and a nucleoside? Yeah, the presence or absence of a phosphate. So what does that teach us? That teaches us that the nucleotide is able to have two different functions. It has the function of storing and providing energy. We use this nucleoside to hang on it the phosphates, and we use this nucleotide to remove the phosphates when we need them. We catabolize, we break down this phosphate bond to get energy, and we build the phosphate bond to store the energy. But we do not only use the nucleotide for energy. What else do we use the nucleotide for? Besides energy, what are the other uses of nucleotides? Yeah, for genetic information. So we have nucleotides. They are very important for energy in the form of ATP or CTP or GTP. And nucleotides are also important for, besides energy, nucleotides are important for genetic information. Genetic information in the form of DNA and RNA. And then we talked about the structure. The structure, yes. Yes.
So you said that as they go, the nitrogenous base would bind to like the third carbon.
Yes.
And the phosphate would bind to the fifth carbon.
Yes.
But like if you look at this picture.
Okay, this is a different picture. When we have the binding of the nucleic acid, we're going to talk about it when we have all the DNA lined up above each other. Then that will make more sense. But now this is a one structure. This is a one nucleotide. So when you look at it holistically, when you look at it like this, so now it will make more sense. Now you have to understand that this is a nucleotide. That is our nitrogenous base. Our nitrogenous base is always bound to what position? Nope. Our nitrogenous base is always bound to 1. That's the oxygen. That's carbon 1. That's carbon 2. That's carbon 3. That's carbon 4. And the top one is carbon 5. Okay? So this carbon 1 is always bound to the nitrogenous base. So as you see here, this is carbon 1 bound to nitrogenous base. That's carbon 2. And then carbon 3 is going to bind the phosphate of the nucleotide below it. So you don't have to know it in that detail. Don't worry about it. You don't have to know it in that detail. But what you need to know is that we have purines and we have pyrimidines. Purines have 2 rings. Pyrimidine has 1 ring. The purines are pure or good. Purines are adenine and guanine. And pyrimidines are, there's 3 of them. There's cytosine, thymine, and uracil. That's what you need to know. What is the name of the bond between the sugars? What is the name of the bond between the sugars called? Glycosidic bond. What's the name of the bond between the amino acids called? Peptide bond. What is the name of the bond between the nucleotides in the DNA called? Yeah, phosphodiester bond. Yeah, this is the stuff that you need to know. You also need to know how many hydrogen bonds are between your A and T. How many hydrogen bonds are between the A and T? Okay, and you need to know the skeleton that there's always a nitrogenous base. Nitrogenous base. It is connected to a sugar, and to the sugar there's a phosphate. And the nitrogenous bases, they could be either A or T or G and C. And we know that A and C are known as purines. We say pure or good. I'm sorry, A and G, pure or good. Purines have two rings. Purines have two rings. And pyrimidines, even though they have the longer name, pyrimidines, even though they have the longer name, they have one ring, they are smaller, and we have three of them. Uracil in the RNA, uracil in the RNA, and thymine, and tyrosine. These are your pyrimidines. How do we bind? A binds with T, G binds with C. What are the kinds of bonds? These are hydrogen bonds. Between A and T, two hydrogen bonds. Between G and C, three hydrogen bonds. And the bonds of the DNA molecules connect vertically through the phosphodiester bond. What kind of bond is the phosphodiester bond? Is it a hydrogen bond or is it a covalent bond? It's a covalent bond. When we heat up the DNA and it denatures, who's broken? The phosphodiester bond or the hydrogen bond? Yeah, these are the things that you need to know. You don't have to know how is it the DNA is assembled. Just appreciate also that the second carbon, this one is very important. Why is this carbon very important? Because if it has an oxygen, it's a ribose, and that's found in the RNA. If it has no oxygen, they devoid, removed, so it's deoxy. So that is found in the DNA. And remember, the DNA is anti-parallel. So it's like two snakes, but one has a head up, tail in the bottom. The other one has a tail up and head in the bottom. And they connect horizontally with hydrogen bond. They connect vertically with a phosphate bond, phosphodiester bond. Okay. Then we know that the gene is a region or a segment of the DNA that codes for a particular protein. So that particular protein is first coded in the form of mRNA, and that mRNA is a nascent. What does nascent mean? Is a nascent transcript. What does nascent transcript mean? Yeah, so that means it's not ready yet, but excellent. It has introns, and exons. It is a combination of introns, and many exons. So we're gonna do surgery to this nascent mRNA, cut out some regions and keep other regions. Who do we cut out and stick together, and who do we keep and keep it as a whole to go from nascent mRNA to mRNA? So who's removed? Excellent. So the introns are removed. They are excised, and exons stay in the mRNA. So remember that this is happening in the nucleus. Inside the nucleus, you have this transcription. So whoever is going to exit the nucleus, it means it's going to go get translated. So if you're going to exit the nucleus, you're going to get translated, you're the exon. So they call them the exons because they're going to exit the nucleus to be translated in the ribosome. So if you exit the nucleus to go get translated, then you are an exon. So exons exit the nucleus to be translated, and introns remain in the nucleus, and they do not get put together except in a concept known as alternative splicing. What is alternative splicing? Excellent. What is alternative splicing? Exactly. So now they start to recycle all of these introns, putting them together and to make some useful genetic information and be able to use them to code for beneficial proteins. We talked about condensed chromatin and that not all the chromatin is being transcribed. There is heterochromatin and there is euchromatin. Euchromatin is what the cell needs, what the cell transcribes and needs. And then there is also the heterochromatin, and that is what is inactive, not being transcribed. And we have to know the structure of our DNA. It is a double helix. It is anti-parallel. The side that has the phosphate group at the top, that is the 5' end. So that shows you this is the 5' end. The side that has the phosphate group is the 5' end. And the side that has the OH group is the 3' end. So they are anti-parallel because one side has the 5' and the other side has the OH group. So if your OH is at the top, that's the 3' end. If the phosphate is at the top, that's the 5' end. What charge is holistically, when we're looking at the DNA, what is the total charge of the DNA? Is it positive or negative, and why? It's negative because of the phosphate group. Excellent. Excellent. Okay. Okay. So because the DNA is negatively charged, who is positively charged and stuck to this DNA very much and is able to coil the DNA around itself and loop it? And the DNA, if it's spread out, it could be as long as an entire football field. But what ties it down into little, very little coils? This is known as a structure known as the nucleosome. So the nucleosome is when the DNA is wrapped tightly and coiled around these proteins known as the histones. So histones are proteins, and their jobs are to round the DNA. They cluster in groups of 8 molecules, and the DNA molecule winds around the cluster, and it creates a granule called the core particle. Chromatin consists of thousands of repeating core particles, and chromatin forms complex loops and coils. Each chromosome is packed into its own region of the nucleus called the chromosome territory. Okay, so we have to also understand that chromatids are joined in the center at a what's known as a centromere. In front of the centromere, there is a plate known as the kinetochore, and these are protein plates or protein plaques onto which when there is a cell division, there are microtubules that attach to the kinetochores, and they are the ones when the sister chromatids line evenly in the middle of the cell plate on the metaphase plate. They are the ones, these microtubules attach to the plaques and the plates known as the kinetochores, and then they pull them and pluck them off, pull them together from that kinetochore plaque, and that is what's gonna lead us from the metaphase into the anaphase. So you see, these are the centromeres, and the kinetochores are the orange plates or the orange plaques. Who attaches to these orange plates and these orange plaques? Is the microtubules. The microtubules attach to these kinetochores, and they pull in opposite direction, and that's what splits for us these sister chromatids in cell division. So in the metaphase plate, all of the sister chromatids are going to be lined up as an X, but then in anaphase, they're going to be pulled from this orange colored plaque or orange color plate. And we talked about ribonucleic acids. They come in many forms with different functions. And the difference between them, and this is very high yield for purposes of examination, and that is the base uracil. Uracil replaces thymine. And we have different types of RNAs. There are microRNAs. There are non-coding RNAs. But the major ones that you need to know are your tRNA, your rRNA, and your mRNA. And they are actually lined up in what order? This order that we see. Messenger RNA the first, ribosomal RNA the second, and transfer RNA the third. What does that represent? Represent size. So the mRNA is the largest and the biggest of all of them, followed by the ribosomal RNA, and then the smallest is the transfer RNA, because its job is just to transfer one amino acid. Okay, so when it comes to the genetic code, the body can make millions of different proteins. So now they study the regions of the DNA that are specialized into making proteins that usually undergo mutation. Like, for example, when it comes to sickle cell anemia, in sickle cell anemia, there is a single nucleotide polymorphism. The single nucleotide polymorphism, SNP, leads to a mutation where instead of us having the required protein, glutamic acid, we end up with a protein, instead of having the proper amino acid, glutamic acid, we end up with a different amino acid known as valine, and that causes a deformity. The deformity is sickle cells will only sickle in sickle cell anemia. They will only sickle when you need them the most, when there is a drop in hydrogen, when there's a drop in oxygen levels, when there is excessive stress, strenuous exercise, and you demand more oxygen, then they start to sickle. So what happened here is that codon, one codon was altered that led to the transcription of a different code that led to a different amino acid. So we transferred from having glutamic acid into having valine. The process of transcription, begins with the start codon. But this specialty of studying the regions of the DNA that are concerned with mutations. So there are particular set of regions in the DNA that undergo mutations that lead to these types of diseases. So this is what's known as genomic medicine, is when they focus on the regions of the DNA that are usually implicated in diseases and mutations, like that problem with sickle cell anemia. The base triplet is a sequence of 3 DNA nucleotides that stands for 1 amino acid. So 3 nucleotides, they are genetic information for 1 amino acid. And the codon represents 3 base sequences in the mRNA. There are about 64 possible codons available to represent the 20 amino acids. And there is a start codon. We have to know that start codon, and we have to know what it also happens at the same time. So this start codon is very special, AUG, because it starts for us the process of transcription, and at the same time it codes for this very important amino acid known as methionine. And there are also stop codons that when we read them, the process of transcription halts and comes to an end. And these are your 3 stop codons, UAG and UGA and UAA. Whenever your ribosome reads one of these 3 stop codons, it will stop the process of transcription or translation. This is when the mRNA, this is when the RNA polymerase is going to stop the process of is it translation or transcription? Translation. We have a start codon with an amino acid, methionine. And then we have a stop codon to stop the process of. Raise your hand if you think it's transcription. Raise your hand if you think it is translation. It is transcription. We are transcribing. This is a start codon to start the process of transcribing an mRNA transcript. And then we stop at that region. We have transcribed all the codons for our necessary structural or functional protein. Are we clear? Yeah. Okay, so we already talked about the central dogma of biology, and we go from DNA to RNA into protein, and this is called transcription. And mRNA protein is called, the mRNA going from mRNA to protein is called translation. And we have to know the processes, and we have to know where they occur. So transcription is always taking place in the nucleus, and translation is happening in the cytoplasm. That's why it's convenient for us to remember that exons exit from the nucleus where they were transcribed so that they can be translated in the cytoplasm. Yeah, in the cytoplasm. Cytoplasm is the region. What's the difference between cytoplasm and cytosol? So the cytosol is just the colloid. So remember that we said there's a difference between solution, suspension, and a colloid. Solution is when you have, what's the difference between solution and suspension? What is the difference between solution and suspension?
So like in suspension, the particles are separated. They may be together with water, but they receive
No. Yes.
Solution
and suspension.
Solution contains solutes.
Yes. Contains suspended stuff. No. What is the answer? What's the answer between, what's the difference, Melissa? Solution and suspension.
Solution is when something is dissolved into a solute, and a suspension is like
suspended. Stuff is suspended.
When it's like when two different, I don't know what to call them, solutions are together but not mixed.
Not true. Yes.
Isn't it when it's just there, but it's not dissolving?
Then that is not, if it's not dissolving, that's like having fat in water. That's an emulsion. So that is not suspension. Okay. They are all dissolving. A suspension, the molecules in suspension are dissolved, and in solution they are also dissolved, but the difference is the size. So NaCl, that's salt, but very tiny, 10 nanometers. And also glucose, that's about 500 nanometers, but that's very small. But if you are really large, like 1000 nanometers, like a red blood cell in the plasma, then that's huge, but it's still dissolved. Red blood cells still dissolved in your plasma. That is majorly water with electrolytes and proteins, but it's dissolved. That's suspension. Then a colloid is what the cytoplasm is. That's the cytosol. It is water and proteins. And a colloid is always what color? White, jelly, cloudy. Yeah, that's a colloid. And in the end, we have emulsion. What's an emulsion? Is when you have a solution, but there is no mixture. There is no Solution is when there is no,
Emotion.
I'm sorry, an emulsion is when there is no solid, no solid. It's like oil vinegar dressing. Okay, it's oil and vinegar. They're dissolved, but there's no solid. Both are liquid. Okay, transcription. Transcription is copying the genetic instruction from DNA to mRNA in the nucleus. It utilizes enzymes called RNA polymerase. Remember, we talked about DNA dependent RNA polymerase. That is not good. What we see here in the slide, this slide is not showing you from the book the detail. It uses an enzyme called RNA polymerase. But what kind of RNA polymerase is it? Excellent. So it's DNA dependent RNA polymerase. Go tell that to the retrovirus that has a reverse transcriptase. That one uses an RNA dependent DNA polymerase. So it reverses from mRNA into DNA. So the first step is transcription, and it binds to, hello? No, sorry. Yeah. I lost track. I thought maybe we were in the wrong room. Okay. It binds to, yes.
I'm sorry, this is just but we just want to clarify. Last week you said exon stay in.
Yeah, exon stay in the, not here. Okay, hold on. Exons stay in the final transcript that will be translated, but the introns stay in the nucleus, but the translation is happening outside. So it's confusing because students think intron meaning they will be in. For the, no, introns, they will be cut out and they will stay in the nucleus. Intron and nucleus in the nucleus. Exons, they will stay in the final transcript, but they will exit to the cytoplasm where they will be translated. Okay? That's not off topic. That's in topic. That's core topic. Okay. Okay. All right. And you know your RNA polymerase, you have to know what is it dependent on, because we have different types of RNA polymerases. It binds to the star sequences in the DNA and it opens up the helix. Opens up the helix. This RNA polymerase opens up the helix and reads the bases from one strand of DNA to build a complementary strand of the mRNA. So now look at the law. This is very important. We are making RNA. So with C we get a G, G we get a C, that's known. But wherever there is a T, we would get an A. But the virus, the retrovirus, when the retrovirus is trying to use RNA-dependent DNA polymerase, when it reads a U group, a U nucleotide in the mRNA, what is it going to put on the other side? It's going to put an A, because now it's making for us DNA. And then when it sees an A in the mRNA, it's not going to make a U, it's going to make a T, because it's now making a DNA template. So it's always important, who are we dependent on? Are we a DNA dependent or RNA dependent? Okay, transcription. So the RNA polymerase rewinds the DNA helix behind it. It allows the gene to be transcribed by several polymerase molecules. And then there is a terminator that stops the sequence at the end of a gene. So now we have an immature pre-mRNA. They didn't put my word, a nascent transcript. That's what it is. That's a nascent transcript. The RNA produced by the transcription undergoes processing. So enzymes within the nucleus remove the introns from the RNA and splice the exons together. And we said alternative splicing is when we recycle. We use all the introns, we stick them up together to make from them some useful forms of proteins. You see, these are my You see these are the A, B, C, D, E, F. These are my exons. And the brown ones are my introns. And we have different types of mRNA. We have the messenger mRNA that carries the code for the nucleus to the cytoplasm. And it has a protein cap that is the recognition site for the ribosome. And there's a transfer RNA. It delivers a single amino acid to the ribosome and contains an anticodon. This anticodon is a series of 3 nucleotides that are complementary to the mRNA codon. So this structure is very important because it participates in the actual translation. So it's not a product of transcription, but it participates in the translation. So it's a transfer, it's a transporter. It delivers a single amino acid to the ribosome, and it has the anticodon, which is a series of nucleotides that are complementary to the mRNA codon. So the mRNA, we have made 3 codons. Now these 3 codons will have to bind the transfer RNA's anticodons. And when they bind together, then the anticodon within your tRNA is bound to an amino acid. So every time we read, every 3 nucleotides that we read in the mRNA, they will have their complementary anticodons. And each anticodon is carrying on its head our amino acid of interest. And we keep on shifting until we make beads. Each bead represents an amino acid. We will see this in a little bit. Ribosomes are the organelles. They are like translation machines. They are reading machines. They are the organelles that read the message and build a peptide chain. They contain a large subunit and a small subunit, and they are free in the cytosol, or they are stuck to the rough endoplasmic reticulum. So why is it that we have ribosomes stuck to the rough endoplasmic reticulum and ribosomes that are free floating? Well, we said if we want to know the difference, we have to look at the similarities. Both of them are ribosomes, so obviously both of them are going to be making proteins, but then that's where the difference is going to be. The proteins that are going to be made in the cytosol, they are going to be for the cell, for local use. But the proteins that are going to be translated by the ribosome attached, by the endoplasmic reticulum attached ribosomes, they are going to be embedded into the rough endoplasmic reticulum to gain a signal peptide sequence. And that signal peptide sequence, whenever that signal peptide sequence is read by the Golgi body, the Golgi body is going to know that this protein is for shipment and for exporting. That's like a barcode. And who gave us that barcode? It was the rough endoplasmic reticulum. So the ribosome make the protein, but it is after they make the protein, they dip it inside the rough endoplasmic reticulum, and the rough endoplasmic reticulum attaches that signal peptide sequence, and that sequence is the barcode that's gonna make the Golgi body. After we translate it, the Golgi body is gonna package it, because that's where we make our vesicles. That in that pita bread looking like, when you have many pita breads stacked up on top of each other, that's your Golgi body. So it has one side, that's the cis side, and the other side, that's the trans side. So the cis side receives the protein, then they are packaged in the middle, then the trans side exports them out to leave the cell through exocytosis. Okay, we talked about that. So this is the transfer RNA. It looks like that. This is the anticodon. So you have your RNA here, your mRNA here, where my mouse is, where the orange is. Then the anticodon will be in the back, and then this is the amino acid accepting end. So the amino acid is in the back here on the head, and this is the tail. So there are three types of steps that we have to go through in order. The first step in translation would be the initiation. Initiation of what? Initiation of translation. So the ribosome assembles with the mRNA in the cytosol. And you have to understand that it is not like one ribosome is doing this procedure. It's not one ribosome who's doing the translation process. It is multiple ribosome units that are translating at the same time. So in initiation, the ribosome assembles with mRNA in the cytosol, and the small subunit binds the leader sequence of the mRNA, while the large subunit joins. Then the AUG codon is reached, and the protein synthesis begins. The tRNA brings the first amino acid, methionine, reading that, methionine. So now we are translating. We began with AUG in transcription, and now the methionine is brought. So is AUG the codon or the anticodon? That's the codon. And the anticodon is going to be, if this is an A, then we're going to have a T. If this is a U, you're going to have an A. If this is G, you're going to have a C. So TAC. So TAC is going to be carrying methionine for us. TAC is going to be carrying methionine and is going to bind AUG. And that's going to be our first start code, our first amino acid in our initiation. Then we have to elongate the chain. So the next tRNA with amino acid arrives, binds to the A site of the ribosome. So the ribosome has different sites. There's the second site known as the A site. The ribosome creates a peptide bond between the first and the second amino acids. The ribosome creates peptide bond between the first and the second amino acids. Ribosome moves down the mRNA by one codon. So the growing peptide is now attached to the tRNA. So the growing peptide is now attached to the tRNA in the P site. And the process continues until the polypeptide grows. In the end, we terminate when we reach our stop codon. And we said our stop codons are, what are our stop codon? UAG and UGA and UAA. You see one of those as termination. Excellent. Making proteins for packaging or export. I'm sorry? What did you guys say?
How you remembered it, you go away.
Oh, I said that?
Yeah, you go away.
I'm surprised how you guys know that. I said that. Oh, wow. Okay, so making proteins for packaging or export. So proteins are destined for lysosomes or secretion are modified by the ER. So the ribosome docks, you see the ribosome docks in the ER. So I mentioned to you that these rough endoplasmic reticulum bound ribosomes make for us proteins that are going outside or to the lysosome. So sometimes we need some enzymes, degradative enzymes, proteolytic enzymes. Those ones have to go inside the lysosome so they can help degrading the microbes. So the lysosome is full of acid, hydrogen proton, it's full of proteolytic enzymes, it's full of metalloproteinases, it's full of proteolytic enzymes, degradative enzymes. These enzymes are proteins that are all made in your rough endoplasmic reticulum bound ribosomes. So the endoplasmic reticulum modifies and packages protein in transport vesicles. And there are multiple ribosomes in clusters known as the polyribosomes. They can translate the same mRNA molecule simultaneously. Okay, you guys see? This is the protein cap that has to be translated, and that's the ribosome. And now you see that multiple different ribosomes are translating. Do you guys see? This is our one mRNA. What does it have? That's the protein cap. And then you see that one ribosome is translating here, second ribosome is translating here, third ribosome is translating here, all these ribosome subunits. Which one binds first? The small subunit binds to the mRNA or the large subunit? Who binds first? No, the small subunit binds first. We mentioned it here. You see? Small subunit, you see? Small subunit binds the leader sequence of mRNA and the large subunit joins. When AUG codon is reached, the protein synthesis begins and tRNA brings first amino acid methionine. So multiple ribosomes in clusters called polyribosomes and translate the same mRNA molecule simultaneously. Okay, so these are the sites we need to know. That's the exit site, that's the P site, the polypeptide site, and that's the A site. Okay, so this is a pool of free amino acids. The tRNA binds an amino acid. So the transfer RNA binds an amino acid, and that costs energy. Just like the sodium potassium ATPase hydrolyze energy and use ATP in order to attach an amino acid. The tRNA delivers the amino acid to the ribosome, and it enters in the A site. What happens now, there is a reversal. You now move down this path, but what happens is the peptide is shifted to, so there was a codon here. You see this one had the MET, the methionine. So methionine was on this one. When it exits, we shift the amino acid to the P site. Then the amino acid that's on the P site shifts to the exit site, while all the two amino acids on top of it shift to this. So now do you see that's histidine, that's alanine, that's methionine. So now when the one on the P site moves to the exit site, the one on the A is going to move to the P site, and it's going to have 3, and so on and so forth. So the tRNAs are shifting left bound. They're going left to the exit site, but the polypeptide chain is shifting to the right. It's going to the A site, and that's how we assemble our beads. So that's the confusing part that students have difficulty with, knowing the direction of the ribosome movement. So the ribosome is moving to the right, creating for us a bead of polypeptides. So these are my peptides, and we are moving to the right, but our transcript is moving to the left, and the tRNA is moving with it. So you have your mRNA that you're reading is moving left. The tRNA is exiting left, but the ribosome is moving right, and the polypeptide is assembled right, to the right direction. We are moving to, so it's EPA. E for exit. We're exiting to the left. The mRNA is moving to the left, but the ribosome is moving to the right, and our polypeptide is assembled to the right. But in the end, when we read the last one, when we hit the stop codon, what's going to happen? The last one, let's say this is now, it's not CAU. This is now UGA. When we hit UGA, we stop. Then the whole polypeptide is now going to shift to the P. Nothing else is going to be added. Then we're going to shift to the E, and it's going to exit. But this is temporarily. This confusing part is temporarily to add me, because one tRNA is leaving and another one is entering. So we add right bound. But when we're finished, then we go to the P site, then the E site, then exit outside. Is that clear? Okay. So this is our tRNA. You see the tRNA? This is our tRNA. The tRNA on its head, it has an amino acid. That's how it is. There is a tRNA that binds to every codon. This is our anticodon. GUA is going to carry for us this amino acid. You see the amino acid. That's how it looks. The tRNA looks like that. On the bottom, it has the anticodon. On the top, it has the amino acid. Okay? And this red part, this pink part, that's your, what is that? Yeah, that's the codon. Excellent. That's the mRNA transcript. All of it, all of the exons. These are all the exons connected together. So what happens is the small subunit binds first. And on top of it, the large subunit is all empty. The E site is the exit site. The P site is the one in the middle. And the A site is the one that is on the rightmost. What happens is we're going to begin with AUG. We're going to begin with AUG. And what happens is your tRNA enters the A site. If they call the A site if they call that entry, it would have been easy, but E begins with exit too, so we cannot have two E's. But think of A as an entry site. So the tRNA enters with its amino acid and anticodon on the A site. Then what happens is we shift to the left on the P site, and we have our amino acid here. Then when we have delivered our amino acid, the tRNA moves to the exit and goes alone outside, leaving behind its amino acid. Then the next tRNA comes in with its ALA, with its new amino acid, alanine. And what's going to happen is the tRNA is going to shift from A to P, then from P to E and exit. But the amino acids are going to move from P to A to the next one. So now you're assembling beads on the top of the next. So each tRNA is going to enter into the A and is going to accept all the amino acids that were made before in the P site. And then the tRNA is going to leave, and then the one in the middle is going to have the peptide chain. Then a fourth one is going to come in, and all the peptide sequence is going to be shifted to it, and the empty one is going to exit to the exit site. And that's how we're going to move. We're going to recycle all of the tRNAs. The empty ones are going to move to the left to the exit site, and the peptide is going to move to the right until we meet our stop codon. When we meet our stop codon, we terminate, and then everybody now is going to move to the left. We're going to move from A to P to E, and then we're going to exit when we terminate. So you just have to read that. It's not that complicated. You just have to read it. It's going to make sense. Yes, you had your hand up.
So every tRNA, once it goes into P, it leaves the amino acid behind?
Yeah, because now it's going to go empty. Imagine I am a tRNA. I walk in holding my amino acid, and I walk right outside with it. I can't. I got to give it to the one behind me. So the one behind me is going to hold my amino acid and his amino acid, and then exit and hand over the torch to the one behind it.
So it's like a relay race.
Okay, I'm sorry. Can we have one person talk at a time? Yes. What did you say? Yeah.
Those two, the purple and the blue, right?
Yeah.
That's the one that this one is getting from the previous.
Yeah, so the one in the middle is blue. The one in the middle is alanine. You see the one that left? That was the start codon, purple. So blue accepted purple on its head, and now it has blue. Then blue is gonna move. Then the empty tRNA is gonna leave, and the one behind it, green, is gonna hold the three in the middle. It's gonna hold on the very top methionine purple, blue, and then on the bottom histidine green, and then it's gonna hand it to the next guy, and then it's gonna go out until we reach the last one. The last one is gonna accept all of them, and it's gonna go out with them, and then they're gonna separate, and then you
Free?
yeah.
Okay,
Free primary sequence. Then it's gonna undergo the hydrogen bonding and going to undergo all the hydrophobic effects and go from primary to secondary. You understand? This is just the little confusing part, is that how we exit and how we make the polypeptide. So the polypeptide temporarily is being carried in the opposite direction from the P to the A, P to the A until we hit the termination sequence. Then we're gonna move in that same direction and exit the ribosome. That's very important. Okay, and now do you see the docking? You have to be able to explain that figure on the question. This is where all figures, no figure left behind. So on the exam, you're gonna see this figure. So now I believe I don't need to make a lab study guide because now you know, you just have to know every single figure.
I still like the study guide.
Okay, okay, I'll make one, no problem. But I'm just gonna copy and paste it. Every single diagram is gonna be there, and you have to just understand. So what are we seeing here? That's a rough endoplasmic reticulum because it has a ribosome embedded in it. So look at the pink mRNA is being translated, but now the polypeptide is not free in the ribosome. It is dipped, it is docked and dipped inside the core of your rough endoplasmic reticulum. Then the rough endoplasmic reticulum is going to add a signal peptide sequence and then remove it in a vesicle. That vesicle is going to go for either the lysosome or the Golgi body. Which side is it going to enter the Golgi body, the cis or the trans side? We enter the cis and we exit the trans. And when we exit the trans, where are we going to head to? We're going to head to the cell membrane. We're going to head to the cell membrane to be sent out the cell. That's the polyribosome. So what is the relationship of a DNA-based sequence to a peptide structure? So the DNA is a double helix and 7 base triplets on the template strand of a DNA and the corresponding codons of the mRNA transcribed from the DNA triplets. So you see, this is my DNA. I transcribed it. Then I transcribed it. So this is my DNA, TAC. I transcribed TAC into AUG. So that's my mRNA. You see, I no longer have A's that are bound to T's, they are bound to U. So that tells you, so on the exam, when you see this figure and there's a number next to this, what is that? You can say that has a U, that's a R, that's a mRNA. What's that? That's tRNA. What is holding? That's our amino acid. MET, methionine, alanine, glycine, threonine, histidine, glutamic acid. And then they are all being translated and they are linked in a peptide chain by what kind of bond here? That's a peptide bond. Okay, so the protein synthesis is not finished when the amino acid sequence, the primary structure, has been assembled. So to function, the protein must fold into precise secondary and tertiary structures. Chaperone proteins guide. So these chaperone proteins have the job of folding the newly synthesized proteins, and they prevent improper association between different proteins. And some are also called stress proteins or heat shock proteins. Chaperones produce in response to heat or shock, and they help damaged proteins fold back into correct functional shapes. What did we say about mad cow disease? What happens in mad cow disease or Creutz's Gefilde disease? So when proteins are misfolded, so everybody has misfolded proteins. You and I currently, it's natural, but just a little bit. So the misfolded proteins, the fidelity of folding proteins is very high in our cells. So we don't have a lot of misfolded proteins, but we do have a little bit, and we have the built-in systems to repair the problem. We do have reactive oxidative species, but we also have the antioxidant built-in system to repair that. We do have misfolded proteins, and we also have the systems that repair the folding. Or if they are damaged beyond repair, then we initiate apoptosis and remove it from the cell. But what happens in alcohol consumption, now the rate of reactive oxidative species is going to be way more than the built-in system. It's going to be overwhelmed. And then these systems are going to defect and the cells are going to are going to be are going to be damaged. Same thing with the misfolded protein. So the misfolded proteins, when they go farther than the capacity and the transport maximum for these repair systems. So what's supposed to happen is that there is a misfolded protein response, an alarm that goes in the cell that tries to utilize these chaperone proteins to refold them properly. But if it cannot happen, then the cell must commit apoptosis. In cocaine addiction, the rate of misfolded proteins in the brain is unbelievable. So that causes so many deposition of misfolded proteins in the brain, and that what causes the brain damage, and that what causes exactly like mad cow disease. That's what happens in mad cow disease. The cow is unable to stand up, and the cocaine addict, after substance dependence and abuse for a long time, they cannot walk, they cannot stand up, and then they start to lose sensory and motor properties because of the misfolded proteins that cannot be removed and properly folded. So these chaperones are very important. Let's stop here, and if you raise your hand if you need more time to work on the labs. Okay, so finish that, and I'll send you guys out. I'll send you the study guide